All right, just to recap, whole genome is fragmented in large, very large chunks. These chunks are cloned into large yeast artificial chromosomes typically, and then they get restriction digested and subcloned, then sequenced, and these sequences are then assembled to reassemble the larger clones, and then the larger clones are pieced together to assemble the larger contig. And so this assembly process relies on the fact that sometimes, because we have multiple copies of the genome, there will be overlapping regions, and so we can say, oh, this region is physically associated with both this region and this region, so we know that we can put the puzzle together and say that this is really what this sequence looks like. Okay, so that was map-based sequencing. Shotgun sequencing, which is what Celler was using, is similar, but faster. And the key is that this assembly process used to take a really long time because the computing power and the algorithms weren't good enough yet. Celler found a way to assemble fragments, very small fragments, without doing this double stage, this two-stage cloning, this cloning then subcloning. And what they did is they fragmented their DNA. They just did one series of cloning into vectors, which typically were BACs, I think, bacterial artificial chromosomes, which can handle larger chunks, but not as large as a yak can. Then they clone these vectors out into bacteria, and then they sequence the inserts in the library, and then they just take those fragments and they assemble them into using regions of overlap to assemble into a larger contiguous fragment or contig. Okay. So this approach requires better computer algorithms for assembly, but it also requires greater depth of coverage, what we call depth of coverage. And what this means is that for this region, we have 1, 2, 3 fragments covering it. So that means that we have 3x coverage. Shotgun sequencing for reliable, for good assembly can require 20x or 50x or even 100x. And so the more depth of coverage you have, the better, and the better able that you are to assemble larger and larger fragments with greater certainty that that's what you're assembling something that is actually real. Okay, so the whole genome shotgun sequencing was way faster than the map-based approach, and Celler was able to complete the human genome in around the same time that the Human Genome Project did. So it required about half the time, and it was also probably less expensive. All right, so both approaches still had to sequence their clones, and the way they did this was through Sanger sequencing. And the way this works is it is similar to the polymerase chain reaction, PCR, that we learned about last time, but with one difference. So recall that DNTPs have an OH group on the 3' carbon that facilitate the formation of the phosphodiester bond between the phosphate on the 5' and the OH on the 3'. If we add our DNTPs, but then we also add some DDNTPs into the mix, and what this means is that this OH group is lacking, so that nothing can be added onto this nucleotide. These DDNTPs, they are dideoxyribonucleotide triphosphates instead of deoxyribonucleotide triphosphates. These DDNTPs, they're going to stop the reaction, and they're going to do it at fairly random time points. And so they're in the mix of DNTPs at a lower concentration. So usually you're going to get DNTPs. But every once in a while you're going to get a DDNTP incorporated into the new strand, and that's going to stop the reaction because there's no OH group to add another nucleotide onto. So these DDNTPs are also labeled with a specific fluorescent dye that's specific to whatever base they're carrying. So here A is green, T is red, G is black, but actually it's yellow, and C is blue. So, so let's say we have some, here's our template. We're going through our PCR reactions, and occasionally we get incorporation of, say, here is a G. And that's going to stop reaction. And it's going to stop the reaction at a very specific size. So some reactions are going to stop after the first nucleotide, some are going to stop after the second, some are going to stop after the third. And so you're going to get continuous range of sizes that differ by a single nucleotide. These fragments are going to be separated using principles very similar to gel electrophoresis. They're going to be separated based on size. And through, rather than doing it through a gel, they're going to do it through a very thin capillary tube, which also separates fragments based on size. And as the fragments go through the tube, they're going to go past a laser detector. And the laser is going to excite the fluorescent label on the ddNTP and detect that color. And so if there's a, let's say, a red light here, that we know that that was a T. If there's a red light for fragments of this size, we know that that was, or sorry, a yellow light, we know that that was a G. If there's a blue light, we know that this is a C. And so the laser is connected to a computer, which records the light, which records the color, and that color is directly representative of the sequence of the DNA. And so that's how Sanger sequencing is done. And I think his name is Fred Sanger, won a Nobel Prize for this. He actually also won a second Nobel Prize in chemistry for another discovery that he made. Okay, next-gen sequencing, we covered that. Okay, so next-gen sequencing. Next-gen sequencing is even cheaper, it's even faster, it's even better than Sanger sequencing, but we do still use Sanger sequencing for certain things. But just to give you a sense, and so all of these techniques, like previous genome sequencing, they use fragmentation. We always have to fragment the DNA. And we can do this either through restriction digestion or through sonication, which is exposing it to high frequency sound waves and physically breaking up the DNA that way. We then have to ligate onto these fragments. We need to ligate adapters. And what these adapters do is they give us a known sequence that allow us to kind of manipulate DNA of different sequences all the same. So what we can also do with these adapters is PCR amplification, because they're essentially now primer binding sites. And so we can amplify the DNA and just make more of it. And then we bind it to some sort of a solid surface. It can be a plate, it can be a glass slide, and you're going to end up with this lawn of single-stranded DNA bound to a solid surface. We then do a sequencing reaction, which is very similar to PCR, and this is like Sanger sequencing also does the sequencing reaction. That's what generates all those fragments of different labels and different sizes. And so we do the sequencing reaction directly on the solid surface to which these single-stranded DNAs are bound. And in the case of Illumina sequencing, this solid surface is a glass slide with 8 channels etched into it. And on the bottom of these channels are a lawn of these primers, essentially. And the primers are a way for the sequencing reaction to take place. And so what happens in the bottom of each of these channels is it's very similar. Let's see if it's very similar to the ddNTP method of Sanger sequencing, but instead of using ddNTPs, they have a special mechanism for removing the stop. So it's not exactly ddNTPs. They stop the reaction. They kind of wash the channel with a mix of ddNTPs, when the next nucleotide is incorporated, it then stops the reaction, and so each new nucleotide has a mechanism for not allowing the reaction to go further. So everything is added a single nucleotide at a time. That stop mechanism is then removed. Another nucleotide can then be added, and so each cycle of addition of a nucleotide occurs one step at a time. And when that happens, you get the laser kind of, or camera records, so a laser excites the fluorescence, a camera records the color, and when you get is a spatial distribution of all these different colors. So we know that on, in this spot, an A was incorporated. On this spot, a G was incorporated. And so there's a computer recording for every spot. I mean, there are millions and millions of spots. We get millions of sequencing reactions happening simultaneously. And so that's why it's faster to do this. Whereas Sanger sequencing can only record a single sequence at a time, and it takes several hours. This records millions of sequences over at a time, and it'll take a week or so to do that, but you get millions and millions of sequences. So let's see. So I'm actually going to post a video online to explain this a bit more than I did, in a bit more detail, and videos are often more useful in illustrating kind of complex processes. So I'm going to move on to PacBio. So what PacBio is this is a new technology. This is what they might call third generation sequencing, whereas Illumina is second generation sequencing. So the PacBio the method consists of these plates, and they're called SMART plates because that stands for single molecule real-time sequencing. And these plates have like a million tiny chambers, and in these chambers is a single molecule of polymerase that's fixed to the bottom, and each well is big enough to only hold a single DNA fragment. So the DNA is made single-stranded, it's fragmented, it's made single-stranded, and it is similarly to the Illumina sequencing, where you're adding, I can't remember if they add one at a time or not, but basically you have I believe the fluorescent label is added not to the base but to the phosphate, or it's added to the phosphate rather than the base, so that when the phosphate is taken off, there's a flash of light, and there's a camera on the bottom that's recording for this entire plate the different color flashes, and so that when a new nucleotide is incorporated, that color is recorded. And because the background within each of these wells is fairly low, PacBio has fairly high accuracy, and because there's a polymerase down here that's just kind of cranking away at this DNA, you can get really long reads. And so the thing about Illumina is that you end up with very short reads, between 50 to 300 at the most nucleotides long of sequence, whereas PacBio can go up to like 18 kb, which is like 18,000 nucleotides, which is crazy. It still kind of blows my mind that we can get sequences that long. And so PacBio is great for sequencing, especially repetitive regions, just because their repetitive regions are much harder to assemble. All your puzzle pieces look the same, so how do you know which goes where? If you have much larger reads, that helps you assemble repetitive regions much more easily. And so PacBio is becoming kind of a really, and so people use PacBio and Illumina together because Illumina has certain advantages over PacBio. PacBio has different advantages over Illumina. So if you combine those two platforms, you actually end up with a much stronger project at the end.